As I told you earlier, the 6th chapter, molecular basis of inheritance, it is a biggest chapter in the second PUC, the 6th chapter. Already explained the part 1 and part 2, part 3 which commences from gene regulation expression, that is in eukaryotic organisms. So it involves, in eukaryotic organisms there is a nuclear membrane, different nucleus, and there is a nuclear membrane also. And here what happens, the first step here, the transcription level, that is the primary transcription. So transcription, what are all the things, the information from DNA will be passed on to mRNA, that is transcription. And then, as I told you earlier, here this is monocistronic mRNAs are produced in eukaryotic organisms, and split genes can be seen. Non-coding genes are called introns and coding genes are called exons. The removal of non-coding genes introns called splicing, that is the second method. After this, the hnRNA produced, heterogeneous nuclear RNA produced, develops into mRNA. This mRNA thus formed comes into cytoplasm through the nuclear pores, and then the last one is a translation, the language of genes converted into language of proteins. This is about the gene regulation expression in case of eukaryotic organisms. So next to this is Lac Operon concept proposed by Jacob and Monod, that is in prokaryotic organisms, that is the metabolism of lactose. Metabolism of lactose, it involves 3 important enzymes: beta-galactosidase, permease, transestylase. These are the 3 enzymes required for the lactose metabolism. Lactose metabolism, that is studied in the Escherichia coli by Jacob and Monod. This is called Lac, that is lactose operon concept. Operon complexes are nothing but the group of genes. It involves promoter gene, inhibitor gene, operator gene, and structural genes like Z, Y, A are the genes which are involved in the operon complex. As it involves the metabolism of lactose, Lac Operon concept proposed by Jacob and Monod. So here there are other different operon complexes like histone operon complex, a tryptophan operon complex, and arabinose operon complex, and valine operon concept. So these are the different operon concepts given in the textbook, not given so far for NEET examination. Hence this is very, very important. And this kind of Lac Operon concept involves 2 steps, that is switch on condition and switch off condition. Switch on condition, switch off condition. To begin with switch off condition, it is the promoter gene and the inhibitor gene. The inhibitor gene produces a protein called a repressor. This repressor binds with the operator gene in the absence of inducer. When it binds with the operator gene, such operator gene will not function. This condition is called switch off condition. And if inducer is present, lactose, even allolactose given in the textbook, that is important for NEET examination or CET, lactose or allolactose, inducer present. Inducer binds with the repressor protein, which is already binds with the operator gene, and it get then the repressor get released as the inducer binds with the repressor gene, forming repressor-inducer complex, and it releases the repressor from the operator gene. Now the operator gene is functional. So this condition is called switch-on condition. This operator gene, it induces promoter gene to release RNA polymerase enzyme. Then the ZYA genes. Z releases one RNA, the Y releases one more, synthesizes one more RNA, and A synthesizes one more RNA. So the RNA, hence as there are many RNAs, this is called polycystic mRNA is developed. That is very important. Polycystic mRNA is from the Z1 mRNA that synthesizes beta-galactosidase, which is required for lactose metabolism, and the another one Y synthesizes that mRNA synthesizes permease, which increases permeability of beta-galactosidase into the cells. This is also very important. And then transacetylase plays an important role in translation, that is the translating the genetic code, that is very important. This is how Lac Operon concept takes place in the Escherichia coli, and as it involves the repressor gene, hence it is called negative regulation, not asked so far. So negative regulation, this is about the Lac Operon concept. And then after this, Human Genome Project. It is an ambitious, very ambitious project. That is the sequencing of human genes, launched in the year in 1990 and completed in the year 2003. For 13 years, the sequencing of human genome started and completed in 13 years. So lot of time required for this Human Genome Project. That is the sequencing of genome of the human beings. And as we know, the human genome consists of 3 into 10 to the power of 9 base pairs. For each base pair, it costs about $3 US dollars, $3. Hence, it is highly expensive. 3 into 9, 10 to the power of 9, and then into the $3, that comes to $9 billion. Hence, this project is the expensive project. And it is also, it involves a lot of work. If one page having 1,000 letters and a book of such each page containing 1,000 letters, 1,000 pages book, in each page 1,000 letters and 1,000 pages book. Such books, 1,000 pages book, each page 1,000 letters. Such books, about 3,300 books are required to store the information of the sequencing of the human beings. It is a cumbersome project, it is a difficult project, and it is highly expensive. It requires a lot of time. Human beings consist of about 25,000 to 30,000 genes. And about 3 billion base pairs are present. So collection of data, and then the other things which are involved in this project are ethical, legal, social issues. So these are the various things which one should look into while the sequencing the human genome. That is also very important. Ethical, legal, social issues should be, one should keep in mind all these issues. These are the things which are required for the Human Genome Project. So Human Genome Project, first in different organisms, the sequence of genes already done, that is in Arabinose, bacteria, Caenorhabditis elegans, so far not asked, and then Drosophila. Easy, one code word kortini, A, B, C, D. Arabinose, bacteria, Caenorhabditis elegans, and then the Drosophila. So in these things already genes sequenced. And then coming to two important methods are involved in this human genome sequencing, that is EST, expressed sequence tags, that is the genes already expressed as RNA. This is very important. Genes already expressed as RNA called expressed sequence tags. And the other method is based on the sequence with function of DNA. Sequence with function of DNA. That is called sequence annotations. Sequence annotations. And because of these things, sequenced in even also YAC and BAC, that is yeast artificial chromosome, bacterial artificial chromosome. And then, so they adopted a method of determination of the sequence of genes, method developed by Sanger. He did the sequence of amino acids in insulin. Same method adopted in case of human genome project. And then, so what are all the things which are important things one should keep in mind in human genome sequencing? That is the human first the chromosome 1 was sequenced in human beings. Chromosome 1 that was completed in the year 2006 May. In the month of May 2006, chromosome 1 sequenced. So far not asked. This is very, very important for your NEET or CET examination. And then next to this, human genome consists of 3,164.7 millions of bases. That is very important. Not the base pairs. And average gene, any human gene, consists of about 3,000 nucleotide bases. Again, not base pairs. The largest gene is dystrophin, already asked. It consists of about 2.4 million nucleotide bases. As I told you earlier, 25 to 30,000 genes are present in case of human beings, and 99.9% of the nitrogen base pairs, nitrogen bases, these base pairs will be same in all human beings. Only it differs by 0.1%, and 50% of these genes are non-functional. Only 2% take part in protein synthesis, and a sequence of only at one base pair of DNA changes, that is called SNPs, single nucleotide polymorphism. And such kind of SNPs in case of human beings are about 1.4 millions. Apart from this, as I told you earlier, the largest chromosome is chromosome 1, which contains 2,968 genes, and the smallest one is chromosome Y, which contains 231 genes. So this information is very important for your NEET and CET examinations. And then next to this, the DNA fingerprinting technique. As I told you earlier, the human genome consists of 3 into 10 to the power of 9 base pairs, and 99.9% is the same. It differs in only 1% differs. And then there are certain unique things one can find out in the human genes. That is, the bulk DNA can be seen, and also small peaks of DNA. These small peaks of DNA and bulk DNA can be separated based on the density gradient. They are separated, and bulk DNA and small peak DNAs are called satellite DNA, and even microsatellites can be seen in case of the human beings. And there is repetitive DNA, the DNA repetitive DNA, satellite DNA, and also inheritable mutation polymorphism. That is, if the frequency of the gene sequencing frequency in the genetic drift, change in the gene frequency is greater than 0.01, it is called inheritable mutation or the polymorphism. So the satellite DNA, and then the polymorphism and inheritable mutations to DNA, and the last one is variable number tandem repeat. That is VNTR. This comes to about 0.1 to 20 kb. That amounts to 0.1 to 20 kb. So for not acid, that is the VNTRs. So all these things should keep in mind coming to the DNA fingerprinting technique before studying the DNA fingerprinting technique. So this DNA fingerprinting technique developed by Alec Jeffrey. It is used to trace the culprits. Number one, culprits by collection of samples. That is the first step, whether the hair, semen, blood smears are present at the place where crime has happened, and the collection of the samples, that is very important. This is the first step involved in the DNA fingerprinting technique. And this technique not only used to trace the culprits, but also pedigree analysis of the various crops, and in case of this also solves the problem of disputed parentage also. And then the first step, as I told you earlier, collection of samples and separation of DNA from the chromosomes by the chemical method, and cutting the DNA segments, knowing the different things like satellite DNA and VNTR, variable number tandem repeat, and any mutation. So such unique things can be we can sequence in the DNA fragments, and placing these DNA fragments on agarose gel medium. And as the DNA is negatively charged, when electricity is applied, then the fragments move towards the anode. Smaller the fragments, farther they move. This is known as gel electrophoresis method, by which the fragments can be separated. And these fragments should be removed and placed on nylon sheet, and then probes are added. Artificially prepared DNA called probes. This is very important. Probes are added and they form big bands. So these DNA copies can be increased by PCR technique, polymerization chain reaction technique, and they can be magnified, and such the DNA fingerprints should be kept aside, and the suspected persons, the cells should be collected as samples, and same procedure should be followed. That is the separation of DNA from the chromosomes and cutting up of DNA fragments, placing them on agarose gel, and then separating the fragments, placing them on nylon sheet, adding probes and bands formed, comparing these bands formed with the already prepared bands, so that we can easily trace the culprits, and it also solves the problems in the disputed parentage. And it is also required for the pedigree analysis of crops. With this I completed, with the part 3, I completed the 6th chapter, molecular basis of inheritance, which is very important and the biggest chapter, very important for both CET and NEET. Don't forget to subscribe for the further chapters, and I will complete all the chapters very soon before you are going to appear for the NEET examination. And the coming Saturday you have this biology CET examination. So all the best. So meanwhile I will also explain few other chapters which are remaining. So for CET, as you know, earlier in the first PC complete chapters will be given, and for the 4 chapters omitted from the second PUC, so that CET will be easy when compared to the NEET. And for NEET examination, all the chapters, all the 38 chapters you are expected to learn. So all the best. Don't forget, please subscribe. Thank you.